Hello everyone. So we are having discussion on transcription mechanism, and in the last lecture I discussed about the structure of RNA polymerase of E. coli and about the sigma subunits, that the E. coli has different sigma subunit. It has seven different sigma subunits, and about the consensus sequences where the sigma 70 subunit binds. So in today's lecture I will discuss the transcription mechanism in more detail. Let's begin with it. So the TTGACA and TATAAT, these are the consensus sequences where sigma 70 subunit binds. But E. coli has seven different sigma subunits, and so different sigma subunits bind to different consensus sequences. So there are different consensus sequences or different promoters for different sigma subunits. For example, the promoters of heat shock genes are different from the promoters of the genes to which the sigma 70 subunit binds. So it is sigma 32 which binds to the promoters of heat shock genes of E. coli. So, these are the two promoters. This is a promoter for genes to which sigma 70 subunit bind, and this is a promoter for gene for the heat shock gene of E. coli. So you can see the difference. The consensus sequences in both are different. So as like that, there are different kinds of genes in E. coli which bind to different sigma subunits. So they have different consensus sequences. Now I will discuss transcription cycle in E. coli. So the transcription in E. coli or any prokaryote is divided into three phases: initiation, elongation, and termination. So I will discuss the steps involved one by one. So what is the first step? First step is formation of RNA polymerase holoenzyme. So in this, the sigma subunit or sigma factor and core enzyme forms a complex. So first step is formation of RNA polymerase holoenzyme. Then the RNA polymerase holoenzyme weakly adheres to the DNA and quickly slides to the long DNA molecule until it reaches the promoter sequence. That means the RNA polymerase holoenzyme binds weakly to the DNA molecule much before the promoter sequence, and then it slides to the DNA molecule until it reaches the promoter sequence. Then in the third step, at the promoter, RNA polymerase binds tightly because of sigma factor, because sigma factor makes specific contacts with the bases of DNA, and then it forms a closed complex. So in third step, RNA polymerase binds tightly to the promoter with the help of sigma factor, making a closed complex. In the fourth step, RNA polymerase opens up the short stretch of DNA about 10 nucleotides long, forming an open complex or transcription bubble. So, in the fourth step, there is formation of transcription bubble where the short stretch of DNA opens up. The RNA polymerase opens up the small stretch of DNA so that the transcription process can be initiated. And also the transcription bubble is stabilized by the sigma factor as it binds to the one of the exposed strands, and the other strand then acts as a template strand. So sigma factor also stabilizes the transcription bubble by binding to one of the exposed strands, the non-template strand, and the other strand is then free for initiation of transcription. Then in the fifth step, the about 10 nucleotides of RNA molecules are then synthesized using a scrunching mechanism. Okay, so let us see what is the scrunching mechanism. In the scrunching mechanism, RNA polymerase remains bound to the promoter. Because of the tight binding of sigma subunit to the DNA, RNA polymerase remains bound to the promoter and pulls the upstream DNA into its active site, thereby expanding the transcription bubble. This process creates considerable stress, and the short RNA molecules are often released or aborted, thereby relieving the stress and forcing the polymerase, which remains in place, to begin the synthesis over again. So what happens in the scrunching mechanism? The sigma factor, as it stabilizes the transcription bubble, it binds very tightly to the exposed strands, and because of that, RNA polymerase remains bound to the promoter and cannot move along the DNA molecules. So the RNA polymerase remains bound to the promoter and it pulls the upstream DNA into its active site. So because of this process, and this is the scrunching process, and because of this scrunching means squeezed into a compact mass. So basically the upstream DNA is squeezed into the active site. It pulls off the DNA molecule into its active site, the upstream DNA into active site, thereby squeezing it. This is called squeezing. And because of this, considerable stress is generated. Because of this, stress is generated, and the short RNA molecules which are synthesized, that are around like 10 nucleotide long, those are aborted. They are released because of the stress, and that leads to, then the enzyme then again starts the transcription process. It again forms the 10 nucleotide long RNA again by pulling the upstream DNA into its active site. This is called scrunching mechanism. So this is also called this kind of RNA synthesis is also called, or this kind of initiation is also called abortive initiation, because the small RNA molecules generated, they are aborted, they are released. I'm going to explain this crunching mechanism by showing you a video on YouTube so that it becomes more clear to you. So the scrunching mechanism is important because the stress generated by the scrunching helps the coenzyme to break free of its interactions with the promoter DNA and discard the sigma factor. So scrunching mechanism is important because it creates a stress that leads to a release of sigma factor from the enzyme, so that the enzyme can move freely on the DNA molecule, carrying out transcription process. Now I am playing a video for you on abortive initiation and scrunching mechanism on YouTube. Let's watch that. The video is little slow and hazy, so yeah, you will need a little patience to watch it till end. But the video is very good. It gives a very clear vision about abortive initiation and scrunching mechanism. So this is DNA helix, and this is RNA polymerase enzyme along with the sigma subunit, the holoenzyme. And these are NTPs, bases for RNA molecule. This is the promoter region. This is the enzyme RNA polymerase, which binds non-specifically much before the promoter region, and then it moves along the DNA strand and halts at the promoter sequence. At the promoter, sigma factor recognizes the site and binds tightly, and it forms a closed complex. Then DNA strand within the complex unwinds and forms transcription bubble, which is also called open complex. Now RNA transcription starts. Here the bases are coming and the small transcript is being formed. The thread is the RNA transcript. Now RNA polymerase do not move along the DNA strand. It remains bound to the promoter because of the sigma subunit, because sigma subunit binds to the DNA very strongly. So RNA polymerase remains bound to the promoter. And because of this, DNA upstream, the upstream DNA is pulled into the active site of the enzyme, which causes stress, and this is called scrunching, the squeezing of upstream DNA into the site of active site. So, okay, I have to go back here a little. So, this causes strain, the scrunching mechanism. Now, to release the strain, the newly formed RNA transcripts, they are released. This is the newly formed RNA transcript is released from the enzyme, and the bubble takes the original shape. The DNA rewind and the bubble takes the original shape. Now this release of the short RNA strand is called abortive initiation. Because RNA strand they are formed to release the stress, they are released from the RNA polymerase, and this is called abortive initiation. Now this abortive initiation can occur a few number of times before the transcription actually begins. Now for transcription to begin, the energy is provided by the scrunching stress. The stress due to scrunching actually releases the sigma factor. Sigma factor is released from the RNA core enzyme, and now RNA polymerase is free to move along the DNA molecule and carrying out elongation. And with this, the elongation continues. So this was a video about the initiation, the abortive initiation scrunching mechanism. I hope it is clear. So the next step is then elongation phase. So in elongation phase, the RNA polymerase moves forward one base pair for every nucleotide edit, and during this process, the transcription bubble continually expands at the front of the polymerase and contracts at the end, at the rear. So the chain elongation continues at the speed of approximately 50 nucleotides per second in case of E. coli. And this elongation continues until the enzyme encounters the second signal called the terminator. So in elongation phase, the polymerase move forward from 5' to 3' direction, or from 3' to 5' direction of the DNA molecule, one base pair for every nucleotide edit, and it moves at a very high speed of approximately 50 nucleotides per second. And it until it encounters a second signal, that is the terminator. So what happens in the seventh step? At the terminator, RNA polymerase halts and releases both the newly made RNA molecule and the DNA template, and the free polymerase core enzyme then reassociates with free sigma factor to form a holoenzyme that can begin the process of transcription again. So this diagram shows the transcription cycle of bacterial RNA polymerase, and in here the first step is association of RNA polymerase core enzyme with sigma factor, forming RNA polymerase holoenzyme. Then the RNA polymerase holoenzyme binds in a non-specific way to the DNA strand and starts sliding through the DNA molecule until it reaches the promoter sequence. So at the promoter sequence, RNA polymerase, the sigma subunit or sigma factor of the RNA polymerase binds to the promoter region tightly and forms a closed complex. Then the RNA polymerase opens up the DNA strands within it, forming open complex, which is also called transcription bubble. Then after a few abortive initiations, the sigma factor is released from the RNA polymerase holoenzyme, and RNA polymerase leave the promoter site and start moving along the DNA strand, and then elongation takes place. So after the elongation phase, there is termination phase. In the termination, so in the termination, there is a terminator region in the present in DNA. When RNA polymerase reaches the terminator or termination region, hairpin formation takes place, which is required for termination of transcription. So in this, the RNA polymerase leaves the DNA strand as well as it releases the RNA transcript. And then this free RNA polymerase core enzyme then again reassociate with the sigma factor and again gets involved in the second round of transcription cycle. So this is the transcription cycle of bacterial RNA polymerase. Coming over to termination mechanisms. So E. coli has two kinds of termination mechanisms. One is row independent and the other is row dependent. So row is a protein having a helicase activity. So I will first discuss row independent mechanism. In row independent termination, the terminators, that are the sequences present at the termination site, these have two distinguishing features. One is the region that produces an RNA transcript with self-complementary sequences, permitting the formation of a hairpin structure before the projected end of the RNA strand. So it has, the terminators have self-complementary sequences. These are the inverted repeats, which when transcribed into RNA molecule that permit the formation of a hairpin structure. So this is one feature. The other feature is highly conserved string of A residues. Just after the inverted repeats, there is a highly conserved string of A residues that are transcribed into U residues near the 3' end of the hairpin. So we can better understand this by looking at this diagram. So this is the DNA template strand having the inverted repeats in the brackets followed by a string of A residues. So just after the inverted repeats, there is a string of A residues here. So when this stretch of DNA is transcribed into RNA, the RNA can attain this stem loop structure. Okay, when you will draw it, you will get to know that when this is transcribed into RNA can have stem loop structure where these inverted repeats, they can form, these are complementary to each other. These inverted repeats are complementary to each other and they can form the stem and stem loop structure is formed. This stem loop or hairpin loop structure is important for disruption of the RNA transcript from the DNA, and that leads to termination of the transcription. So, the formation of hairpin structure in the RNA disrupts the several AU base pairs in the DNA RNA hybrid segment and may disrupt the important interactions between RNA and the RNA polymerase, facilitating dissociation of the transcript. So we can understand this by looking at this figure. So here you can see the RNA polymerase is bound to DNA molecule, and there is a RNA transcript. This is this green color, this is the RNA transcript, and this is the stem loop structure, the hairpin loop structure, and just after that there is a AU. This is the RNA DNA hybrid having AU bonding. Okay, AU base pairs are there just after the hair loop structure, and this hairpin loop facilitate the disruption of this AU bonding. See, AU bonding is unusual bonding. This is not according to Watson-Crick base pairing, so it is a unusual bonding, and thus this is very unstable bonding. So the hairpin loop structure which is formed here causes the disruption of this AU bonding, and that leads to the release of this RNA transcript from the DNA molecule and the RNA polymerase molecule also. And this is and somehow this also, it is not clear, but somehow this hair loop structure also causes a release of RNA polymerase from the DNA strand also. So this was about row independent mechanism of termination. Now, so in row dependent terminators, they lack the string of A residues, unlike row independent terminators. They have string of A residues, but in row dependent terminators, they do not have string of A residues, but they do have inverted repeats. So stem loop or hairpin structure is formed here. So the row protein is a hexamer having ATP dependent RNA DNA helicase activity. So what happens in row dependent termination? The row protein binds to the RNA transcript, perhaps during early elongation phase, and moves along the transcript in 5' to 3' direction. When it reaches the RNA DNA hybrid within the transcription complex, it breaks the bond between RNA DNA hybrid, thereby releasing the RNA transcript. So we can understand this better by looking at this diagram. So this is a transcription complex. This is RNA transcript, and this is a hairpin loop structure, and this is the RNA DNA hybrid. So row protein binds to the RNA transcript and it moves towards the 3' end, and as it moves along the RNA transcript, it breaks the complementary bonding. It disrupts the stem loop structure, and when it reaches the DNA RNA hybrid, it also breaks the bond between the DNA RNA hybrid, thereby releasing the RNA transcript from the transcription complex. So the row dependent the row dependent termination mechanism requires ATP hydrolysis, unlike row independent mechanism which do not require ATP hydrolysis, but that is a kind of physical disruption. So I hope this is clear to you, and this is it for today. So if you have any query, please write to me on the comment box or in the Google Classroom. Thanks and bye for now.